Thanks a lot, Susan. When I first got invited to this meeting, I was actually really excited to hear that Susan was going to be introducing me because I was a huge fan of Genome magazine. So if anybody hasn't read it, or if there's anyone out there who's wanted to support a magazine, as a genomic scientist, I found it fantastic. So I would really recommend looking for back issues. I don't know if you even access those. You can get access to those. Oh, excellent. Oh, so let's drop by and get them out of the trunk of your car. Okay, so as Susan mentioned, I mean, she told a very compelling story and something that was very important, something that was very close to her heart and very important. I'm going to switch gears a little bit, and I'm going to be talking about things that people use every day and talk about consumer genomics. I'm going to first define how we really are defining consumer genomics, and then I'm going to give a few examples of how we're using consumer genomics to improve consumer products day in and day out. So let me see. As a genomic scientist, and I started in this field, I started at P&G in 1993, so I'm a real old-timer in this area now. But I've watched the whole personalized medicine field really evolve over time. And I've gone to a lot of personalized medicine meetings. The term consumer genomics has started to really go into these kinds of meetings. One of the things that I really noticed as someone who works at a consumer products company, however, is that every time someone talked about consumer genomics, they would use the word patients. Working in consumer products, we don't actually deal with patients. We deal with consumers. And you know, there's a lot of differences between how a consumer interacts with a product and how a patient interacts with a product. Obviously, price is often a factor. There's high volume and low margin kinds of issues that we have to deal with, although there are room for instance, high value products. And of course, it's a different regulatory environment, right? Instead of having the FDA looking over our shoulders, there's opportunities for fads, and there's opportunities to turn this into marketing, snake oil, so things that could be really problematic. The nice thing is those fads are possible, but they're really not sustainable if you want to continue to sell your products. So the most important difference between consumers and patients is that consumers choose. And if I'm very, very lucky, they choose from some of these products. So these are consumer products that we sell. They are just a few of our over 300 brands. 22 of these brands actually bring us over $1 billion US in sales each. And these products really have two things in common. Number one, if you look at them, they all interact with the epithelium, either the skin on the inside of you or the skin on the outside of you. And number two, there's a whole lot of these that really have an interaction with what's known as the microbiome, and that's the bacteria, the fungi, the microbes that either live in, on, and around you. And so we came to the conclusion if we had a really deep understanding of those aspects, we would come up with better consumer products. So let's go in, define how we define consumer genomics. Now, when people think of consumer genomics, a lot of times they think of just the DNA aspect of it, looking at the genetic differences like a 23andMe test. But we have a much more comprehensive definition of what that represents. And to give you an idea of this, I'm going to take you to a cell. Let's say that this is a cell on your skin. Every time your cell is hit by an external stimulus, for instance, you go out into the sun or you put lotion on, there is a programmed series of events that sends a signal from the outside of that cell to the inside of the cell. And that signal, that's known as signal transduction. And actually, when I was in graduate school, I studied that a lot. So that was something that was very interesting to me. But the whole purpose of that signal transduction pathway is to tell the nucleus to respond to the outside of that cell. And the nucleus responds by turning up and turning down genes. And those genes get turned up and down in basically what is a symphony. It's a very complex web of interactions that take place. The genes the DNA is turned into the RNA. RNA is ultimately turned into proteins. And the proteins are the things that go to work inside of the cell and actually make things happen. So for instance, you get a sunburn, protein, and you get some DNA damage, proteins will actually turn up that will repair the DNA damage in your cells. It'll also produce melanin, which will help to reduce additional sun damage. So those chemicals, those proteins go to work inside of the cell and do chemical reactions in the cell. And the byproduct of those chemical reactions are these chemical metabolites. So interestingly, In the field of molecular biology, we take a look at this and we classify these all as omics technologies. So when we're looking at just the DNA sequence itself, that's really genetics, and we're really interested in that aspect as well. When we're looking at the regulation of the genes, that's what we're calling genomics, and that's an area that we spend quite a bit of time. We're looking at understanding how those genes are turned up and how those genes are turned down. We also have people in our company that look at the proteins, and that's called proteomics, and also people looking at those metabolites. That's called metabolomics, and that's an area that's classically known as the omics field. We also play in an area known as the metagenome. I mentioned to you one of the side things that we learned about all of our products is they have an impact on the microbiome, or those bugs that live in, on, and around you. Well, this was actually a picture from 2008 Science magazine that introduced the Human Microbiome Project, where people were just beginning to define all of those bugs that are responsible for your health. And they chose to do their first look at the nasal, oral, skin, gastrointestinal, and urogenital. Well, if you're from P&G, see if this clicks, you're going to find that we have products for all of those. So it's really important, again, that we play a role in understanding this very, very deeply and at a real deep molecular level. So, you know, we talk about all of these things, understanding the regulation of the genes, the RNA. We talk about the metabolites. We talk about the proteins. We talk about understanding the bugs around you. This is a much more comprehensive approach, and the reason that we do this, and we've always been very interested in DNA, and I'll talk a little bit about some of the DNA work we've done. DNA really sets the stage, but it really is not the entire story. What we really need to do is gain a deeper understanding of how your environment and your lifestyle impacts your genes. And so, you know, ultimately DNA is not your destiny. How you use your DNA and what you do with your DNA really does have an impact on what you do. It just sets the stage for where you're ultimately going. So we use genomics in our company really to gain an understanding of genetic predispositions, what happens when you use commercial products, technologies, and ingredients, what happens when you're exposed to your environment, you know, pollutants, the sun, etc. What happens over time? What happens when you age? What happens to your skin as you age? What happens when you're interacting with your microbiome? Also, how do you impact your microbiome as well? So those are all important questions that we're interested in doing. That ultimately leads to the generation of big data. And it was very exciting to see some of the talks earlier today talking about AI and machine learning, because we have bioinformaticists that do quite a bit of this. And we use that big data to help us to identify new ingredients that ultimately go into new products. We do it to support claims and come up with new insights on the existing products that we have. And we also use it, of course, to build intellectual property, and we also use it to build credibility. We present this stuff at scientific meetings, and we show that this is scientifically valid work. It's not just a marketing gimmick. So here's a few specific examples I wanted to go through. I'll spend a little bit of time talking about the skin, and then I'll just touch on some of the other areas that we worked on: scalp, respiratory, oral, and gastrointestinal. So we really, at P&G, were a very early adopter of genomics to understand skin aging. As a matter of fact, when I first started working at P&G, the Olay group was really the people who were very excited about trying this out and seeing what they could do with it. And so we did many experiments that were designed to look at what happens functionally to aging skin. And we mapped gene expression profiles of young versus old skin and try to gain an understanding at the fundamental level of what those differences are. And if we can actually understand those, we can hopefully reverse some of those pathways that are involved in aging. So one of the examples that we did is a collaboration that we did with Harvard University and also with the consumer genomics company 23andMe. That's known as the Multi-Decade Ethnicity Study. In this study, we took groups of women in their 20s, 30s, 40s, 50s, and 60s who were Caucasian, Latino, African American, and Asian. And we did a massive genomic study. We took DNA, we took gene expression samples from various parts of their skin. We did a lot of other work as well, and we basically ran over 6,000 genomic samples to try to identify what happens when you go through the aging process. And we actually found that in the different stages of your life, there are different cellular pathways that are important for that aging process. So if you're in your 20s, you're having issues with natural oxidancy, you're actually not going to be doing very, very well in aging skin. But if in your 50s, your skin barrier begins to break down. So this is kind of a biological process of the aging that happens as you go from decade to decade. And we took all of those genes and we developed an understanding of those processes and said, okay, well, if these genes are actually lower in younger skin and these gene pathways are actually higher in younger skin, can we use ingredients to actually reverse those pathways? And we were able to identify a number of consumer-relevant ingredients. These are cosmetic-grade ingredients that actually regulate these defective pathways in the skin. Of course, that's not enough. If we're able to show that, it's really just the start. One of my collaborators, Zach Kohani, also from Harvard, had a great quote in the Boston Globe in an article talking about consumer skincare, genomics and skincare, and he said, just because you're changing the expression doesn't really mean that that'll prevent wrinkling. So what we do is we actually take it all the way through. We identify the key molecular changes, we identify those potential ingredients, those ones I showed you in that path, we then test for that activity in vitro, but more importantly, we then take it to a clinical study where we can actually prove that it happens. And that's one example of this is we published in the British Journal of Dermatology is one of our Olay products, Olay Pro X, which really did show that the ingredients that we use genomics to help identify really had a positive impact and really had similar impact to tretinoin, an RX grade material. Interestingly, that led to a lot of conversation, right? So if our stuff is so powerful, how do we do this without an RX? Well, we basically have been able to argue that it's because of the ingredients. The ingredients have this long history of human exposure. There's no need for an RX form, so we've been able to really push that forward. And that was really exciting to me because this is one of the first examples. You know, I started a while ago in this field, but we were able to show we were able to identify genes, identify ingredients, and then ultimately make a product that really helps to improve skin. So we've done this with a lot of other areas. One of the areas that we work in a lot is in dandruff, and we're really gaining a deeper understanding of what happens when you have dandruff and what the differences are, the genomic differences between people who have dandruff and people who don't. This is actually a very interesting area for the microbiome because the microbiome plays a significant role in the dandruff. We've also looked at understanding what happens when you're infected with rhinovirus, and this is important for our Vicks brand. This is a great one. We actually paid University of Virginia students beer money to infect them with rhinovirus, and then we scraped their nasal passages to see what happened during the progression of rhinovirus and as the rhinovirus went away. And so that was a really exciting, kind of fun area to work in. We also did something with understanding the transcriptome of gingivitis, which is important for our Crest and our Oral-B brands. What we did was we put a block on your gums. After cleaning your teeth, you put a block on your gums so you can't brush your teeth for several months. So you end up with a really bad case of gingivitis. It's really nasty, actually. And then we take the block off, and we take samples, and we watch the progression of the gingivitis at a molecular level. And then finally, as one of our final examples is gut health applications. And this was really understanding what happens in your gut when you have irritable bowel syndrome. So really, where are we going with this? Well, first of all, you know, we've heard a lot of talk about the $1,000 genome. And there's also been talk about the $100 genome. And George Church, one of the big scientists in this field, is really talking about the $0 genome. What happens when everybody gets sequenced? Well, the interest is likely to extend well beyond healthcare. It's really going to impact healthcare, but it's going to extend beyond that. And what role can we actually play in improving that? And then, you know, obviously, we are gaining, just now at the very cusp of learning more about the microorganisms that are in, on, and around us. And there are a number of microbiome sequencing projects, and we really are gaining a deeper understanding of the host microbiome interactions and how we help each other. And then the real question is, how will the consumer respond? You know, is this really something that they're interested in? Do they just not want to even know about it? We're very interested in understanding that. We do a lot of interviews with consumers on that area. And then finally, how do regulatory organizations respond? We're entering a very interesting field right now where this could be just some kind of marketing gimmick that really isn't scientifically valid. And the organizations like the Federal Trade Commission can step in and all this. So we have really tried to focus on ensuring that we have validated, credible results that other people in the field are capable of doing. So wrapping it up, there's a world of biology outside of pharma. And these areas will be continued to be impacted by the advances in genomics technologies. And it's finally, you know, leaving it with a final thought, it's really critical to maintain the same level of scientific rigor that you see in the pharma field, also in this field. Because good science leads to good consumer products, bad science leads to snake oil, and it ultimately negatively impacts the entire field altogether. We don't want to be in this kind of situation, right, where it's just photoshopping. So with that, I'd like to thank you, and moving on over. Thank you.